my name is Rani, Shah, and today we're going to be talking about the Hershey and Chase bacteriophage experiment. A little bit of background. In 1952, Hershey and Chase set out to test if genetic material was DNA or was protein. And in 1944, Avery, McLeod, and McCarthy proposed that the idea that genetic material may be DNA. But were they really right? So Hershey and Chase wanted to test out to see if they were. The connection between the experiment that they conducted in our exam 2 and 3 material is that in exam 2, we learned that phages can infect genetic information into bacterial cells and then replicate, which is the process that Hershey and Chase went by. And in exam 3, we learned that the processes of genetic material follow replication, transcription, and translation. And one key question that relates to exam 3 material and that's relevant to this experiment is what type of strand is needed for replication. And we'll explore this question at the end. To start out with the experimental design, we had set 1 and set 2, or they had set 1 and set 2. Set 1 contained the bacteriophage coat protein labeled with S35. S35 can only be labeled by protein, but not by DNA. And in set 2, the bacteriophage DNA was labeled with P32, and P32 can only be labeled by DNA and not by protein. S35 and P32 are both shown in color, S35 in green and P32 in orange, because they can show radioactivity. Then both sets were allowed for infection to occur, and both sets had the bacteriophage inject their genetic material inside the bacterial cell. And from there, they took the bacteriophage and bacterial cell and put it into a blender, and were able to blend it up. And of course, theirs was mainly for the lab, and they were able to separate the bacteriophage from the bacterial cells in set 1 and set 2. And from there, they took it and put it in a centrifuge, were able to centrifuge it down in both set 1 and set 2 and form a pellet of bacterial cells at the bottom and the bacteriophage with the liquid at the top. And from that, they were also able to get some cool key findings and results. In set 1, they were able to find out that S35 radioactivity was shown and found in the bacteriophage protein coat, just like the beginning of the experiment, like shown here, where the green is in the bacteriophage. In set 2, P32 radioactivity was found in the bacterial cells and bacterial pellet, shown at the bottom in the pellet form. Now, knowing that genetic material is injected in bacterial cells, like over here in this process that we showed, that genetic information is injected in bacterial cells, and that DNA labeled with P32 is found in bacterial cells, like in set 2, we can conclude genetic material is DNA. And we can also conclude that what type of strand is needed for replication? DNA.